My name is Jie Zheng. I'm an assistant professor from Nanyang Technological University, Singapore. Thanks for the opportunity. So this is joint work with our collaborators over here. And our poster number is 059, so I presented this afternoon. So you're welcome to attend it. So in this work, we try to present a method that combines the knowledge with the data in the modeling of the signaling pathway in cancer. So this is the most important slides in the talk. So nowadays, there's a lot of talks and emphasis about big data, right? This data-driven methods is really prevalent in the field. However, we would like to advocate the importance of knowledge. So there are basically two types of modeling for in systems biology. One is knowledge-driven methods. So this method is to start from prior knowledge and construct the model based on the literature, such as the differential equation or logical rules, and then do the simulation. And this advantage of this method is that it can take advantage of the resources like literature, existing databases, and also it can be very clear in the mechanism, right? What's going on physically in the system. However, the downside, the limitation is that the generic models based on knowledge may not be specific to the context, such as the different diseases, different cell lines. So another limitation is that oftentimes the mechanism and the parameters may not be clear. So it's not easy to build a good model just based on the knowledge. So that's where the data is important. So data-driven methods is to take advantage of the data, so it can allow us to do this context-specific models that specific to a disease. And also, of course, as we know, new data can give us new insights that is not available in the existing knowledge. However, the limitation of data-driven methods is that it can be prone to the missing data or noise, and this is a very important issue. And also, because data-driven methods often is a black box, so it's hard to interpret the mechanism insights. So still we don't know what's going on, even though that we can capture some patterns in the data. And therefore, we advocate the hybrid modeling, which will combine the strengths of these two approaches. So basically, is to have two steps. One is to build the model based on knowledge, that is knowledge-driven. And then we use the data to calibrate the model. So in a sense, the knowledge-driven method is deductive, and data-driven is inductive. So if we can connect them, then there's a cycle, so we can do the iterative modeling. Okay, so we apply this approach to this specific signaling pathway of cancer. So we have this pipeline of 4 stages. First, we build this network model from the knowledge, and then we use fuzzy logic model to simulate the phosphorylation. And then we compare the simulated data with the real data and try to optimize the parameter and the structure of the model so that they can fit with each other well. And then we can infer the network, the learned network. So here is we use fuzzy logic to simulate the data using the Hill function and also the logical combinations of different rules. And then we formulate this problem as the optimization problem of nonlinear integer programming. So here we use a loss function to compare the simulated and the real data. And also we have a balance between the goodness of fit and also the model complexity. We don't want to make the model that's too complex that will have an overfitting issue. So this is a balanced objective function for that learning. So we apply these methods to real data, that is stream 4 challenge. And then we compare with another hybrid modeling methods, Iduati, which is a top performer in this competition. So we have a less complex model so that we have less edges, but we have a better goodness of fit. And this is mainly due to that we take carefully into account the prior knowledge. We are more, have more information from prior knowledge. And then we apply these methods also to the single cell time series data of the cancer phosphorylation and cell fate. So here we have this prior network model, and then we take the phosphoproteomics data, and then we train it the model trained based on the phosphorylation data, and then we predict the cell fate of the cancer cells. So we can see that after training, the prediction of cell fate is better. I'm done. So I'd like to thank the colleagues for sharing the experiences and also the funding agents. And our poster is 059, so you're welcome to stop by. And thank you very much for your attention.